So I'm going to go the next half hour or so, give you a whistle-stop tour of the UK Biobank story and how it's really starting to shape how we do public health research, not just in the UK, but around the world, and how it's starting to transform health. So I'm Naomi Allen, Chief Scientist for UK Biobank, and I'm based in the University of Oxford. So often when I talk about UK Biobank, a lot of people have this perception that we're essentially a fancy biorepository. So we have a nice fancy freezer that holds millions of samples, and we're essentially that we're a biobank. But actually, the reality is something quite different. So I would argue at the moment, we're probably the world's preeminent biomedical resource to do health-related research. We're certainly the largest open access, readily available health research resource in the world so far. We've got the largest repository of sequence data, which I'll talk about later, with the largest data collection of multimodal MRI scans and the largest collection of biomarker data, and one of the largest prospective studies ever taken. So the reality really is quite something different. We're actually a prospective cohort study with a very large, complex data generation scheme on all of our participants, both at recruitment and over time. So before I get into UK Biobank per se, I just kind of wanted to give you a bit of context about the rationale for setting up large-scale prospective cohorts, not just in the UK, but in the US and around the world. So one of the real advantages of setting up a cohort or prospective study is that you can measure exposures or risk factors long before a disease develops, helping to enable researchers to assess whether exposure happened before the disease. So you can assess the temporality of an association. And you can also investigate potential causes of disease among those who go on to develop the disease, compared with those who do not go on to develop the disease within the same study population, thereby minimizing different types of biases. And perhaps lesser known, you can also investigate the effects of an exposure. In this case, let's take something really simple like smoking. You can investigate what are the effects of smoking on a whole range of different diseases, ranging from lung disease to cancer to vascular disease, dementia, migraines, and so on, and vice versa. You can look at many exposures, say a whole range of genetic variants on one disease. So it really gives you a very broad platform in which to do research of risk factors on disease outcomes. The problem with prospective studies is they need to be really large and they need to be long-term, since only a small proportion of the participants will develop any given condition even during a 20, 30-year prolonged follow-up. So this will help us really emphasize why prospective studies need to be so large. So this is data taken from a collaborative meta-analysis, which essentially looked at the relationship between systolic blood pressure, you can see on the x-axis, and the risk of coronary heart disease in different age groups. If you look at 5,000 people, you can see that you can't really tell what's going on. The relationship between blood pressure and cardiovascular disease against different age groups, it's all over the place. If we look at this association in 50,000 people, you can start to see that there, may be some trends between blood pressure and heart disease across different age groups, but the confidence intervals are really wide. You can't really come up with some firm conclusions. Once you get to 500,000 individuals, it's like looking down a microscope. You can see that suddenly things snap into focus. So it's only when you have really large numbers of individuals that you've got enough power in order to look at common exposures with common diseases across different age groups. So that's really why you need very large numbers of individuals to study the relationship between risk factors and diseases, even common risk factors and common diseases in different groups. So UK Biobank was established about 20 years ago now. We started recruitment in 2006. to 2010, and we recruited about 500,000 individuals throughout the UK in 22 different assessment centres, which are located there on the map, across England, Wales and Scotland. And those 22 sites were chosen because they had a different mix of urban, rural, different ethnicities, different social circumstances, and so on. And we collected a whole wide range of information on those individuals. And we now have over 10,000 variables in the UK Biobank database. And crucially, at recruitment, we obtained explicit consent from all 500,000 individuals to follow their health over time through linkage to their health-related records, and also to get in touch with them again for further data collection and assessments. So this really gives a snapshot of, just to showcase the real breadth of data that we have in the resource. So at recruitment, we collected lifestyle information through questionnaires on all 500,000 participants, and we continue to send out web-based questionnaires over time to our participants as well. We've collected blood samples, so we have biomarkers on all 500,000 individuals. I'll go into that in a bit more detail later. We have an imaging sub-study, so we have MRI scans on up to 100,000 individuals. We have genomic data on all 500,000, physical measures on all 500,000, and of course, follow-up through linkage to health records on all 500,000. So a really comprehensive database with a wide range of different types of data that's been collected. So just to go into a bit more detail about the types of genomic data that we have collected. So from the very outset, our funders were very clear that they wanted UK Biobank to be a resource to enable to look at both the genomic and the environmental and lifestyle determinants of disease. So we developed a custom-built genotyping array, which had about 8,500 genetic variants on it, which was then imputed to over 90 million variants. That data was made available for all 500,000 participants seems a long time ago now, in 2017. And I think I can honestly say that the release of that data led to a real step change, both in the number of researchers who were accessing the data, it just overnight exponential increase, but also it led to a real sea change in the field of genetic epidemiology. So anybody who does genetics in an epidemiological context has almost certainly used UK Biobank data, or certainly heard of UK Biobank data. It led to a splurge of GWAS results, of hundreds of traits that are now publicly available on online search engines that have been produced by the Broad, by Stanford, and places in the UK. And all of this kind of standardized GWAS analysis was made possible because of its large size, the fact that we had standardized health outcomes and exposures, and the follow-up through linkage to electronic records. So before UK Biobank, if you remember, we had to do lots of meta-analysis of different case control studies in order to look at genetic variations in relation to disease. With the release of these data, essentially meant you could just use one study, the UK Biobank study, to perform really well-powered GWAS. So the GWAS data really led to the development and wide scale of application of polygenic risk scores for a whole range of different conditions. Now, in essence, polygenic risk scores is essentially taking the risk of a disease related to the genetic variation across your whole genome, not just one gene. And so when you look at genetic variation across your whole gene, what you can see, and this is done by researchers done from Genomics PLC, a company in the UK, what you can see is that for most conditions, there are a small proportion of the population who are at a very high risk, very high genetic risk of developing a condition. Let's take breast cancer, which is the first example there in the top left graph. You can see that about top 3% of high genetic risk of developing breast cancer, about 25% the population will develop breast cancer who will have that genetic risk score, compared with a normal population, which is in the blue line. And of course, there are some individuals who are lucky enough to have genetic variants, which mean they have a very low genetic risk of developing a certain condition. And of course, those with a very high polygenic risk score for developing a certain disease, that's often equivalent to many monogenic disorders. And so the idea has been that, well, we could use polygenic risk scores to enable the early detection and perhaps could lead to screening programs for people who are high risk of developing certain conditions. But of course, there remains outstanding questions on the clinical utility. So, for example, if we all went to the doctor tomorrow and had our polygenic risk scores for heart disease, how does that add onto our age, gender, blood pressure, cholesterol? What does it add in terms of prediction of whether we will actually develop heart disease? So there are questions on your clinical utility, and there are certainly studies in the UK being done now to assess that very question. But it's certainly been an area of high interest in terms of precision medicine. The use of polygenic risk scores has also been used for looking at what's the impact on differential disease diagnosis in clinical care. So this is an example taken from a research group in Exeter, actually, in the UK. This was published a few years ago now, but what they found was that you can use polygenic risk scores to differentiate between those who are at high risk of developing type 1 versus type 2 diabetes, which of course etiologically quite distinct. And you can see from this histogram that the polygenic risk profile is quite different between type 1 and type 2. And what they found was that almost half of individuals, about 40% of individuals with a high genetic risk of developing type 1 diabetes were actually diagnosed erroneously with type 2 diabetes in their 30s, 40s, 50s, and 60s, because that is more common. So they're being incorrectly diagnosed and they're being incorrectly treated. So this has started to lead to a much greater awareness amongst clinicians that actually, if you have a patient coming to you who you think's got diabetes, but who is not responding well to type 2 diabetes treatment, then it may actually be worth considering whether they have type 1. So it's a good use case of how the genetic data can be used to help to differentiate disease diagnosis. Of course, there's much we still don't understand about the role of genetic variation across the whole genome on our health. And a few years ago, a consortium of big pharma companies led by Regeneron invested in UK Biobank to perform whole exome sequencing on all 500,000 participants. So data on the first 50,000 was released a few years ago. Data on the full cohort was released in 2022. And that has really starting to accelerate drug discovery pipelines, which is why big pharma are interested in having these data in the first place, particularly by identifying loss of function variants that are associated with a phenotype that could be used for a drug target. So this is a really good example published by Regeneron in 2021, which I've highlighted the gene of interest here in the red circle, GPR75. What they found was that a rare variant in that gene is strongly associated with low body weight. And because it's a loss of function variant, they were able to use knockout mice models to mimic the effect of this genetic variant on the phenotype. And that's led to real interest in, can you use these genetic variants that have a strong impact on the phenotype to really accelerate drug discovery, particularly around loss of function, where there's a strong effect on the phenotype of interest. So you can already see how these data are starting to be used for drug discovery pipelines. And then, of course, from the whole exome sequencing, we moved on very quickly to whole genome sequencing. And the rationale for having whole genome sequencing data is that it can help us to much better identify disease mechanisms. So how do variation in the non-coding parts of the genome, how does that regulate expression of proteins? How does it provide insights into how diseases develop on a much more biological level? It will also facilitate drug discovery because it will help to identify rare variants that contribute to disease onset and progression. And it will also help us to, in a much more granular way, stratify individuals based on high genetic risk. So like the polygenic risk score idea, but at a much more granular level because you've got detailed information on variation across the entire genome. And it will also enable researchers to look at, okay, can we identify individuals at a high genetic risk for, say, targeted screening, but also which population based on their genetic profile are more or less likely to respond to certain treatments. So this was a public-private partnership between industry, the government, and charities. The first data for the first 200,000 individuals was made available end of 2021, and data for the full cohort was made available a few months ago. So, I mean, I probably don't need to tell you guys this, but, you know, the release of these whole genome sequencing data was a really big deal for us. It's you know, nothing has been released at this scale before, so we're really excited to be able to deliver this project, especially through the pandemic, on time and on budget. And these data, together with all of the other data that we have in the resource, was really starting to transform the way that we can look at the genetic and environmental and lifestyle determinants of disease. So in addition to the genomic data from the blood samples, we've also have focused on how can we turn these limited and depletable samples that we have into really useful, reusable data. So as well as the genes, we've also performed a whole range of biochemistry biomarkers on everyone from the baseline blood samples. And these are the key markers that are of broad interest to most researchers. So things like cholesterol, triglycerides, sex hormones, kidney, liver function, and so on. We've also measured telomere length on all individuals as a biological marker of aging. And we've also done metabolomics using an NMR approach. This is from Nightingale Health on all 500,000 participants, which is mainly a lipid platform with some amino acids. And we've done a pilot study of proteomics on about 57,000 participants. So about 10% of the cohort, using 3,000 proteins of the Olink platform, which is already starting to provide some really useful information. But before I go on to that, I just wanted to kind of rehearse with you why we've taken the approach we have on doing cohort-wide sample assays, because very few other studies do this. Most studies take a case-controlled approach, where you measure a biomarker in those individuals who have developed the disease and a matched group of those individuals who haven't developed the disease. The problem with this approach is that it can deplete the sample very rapidly, and it may yield assay data that's not comparable when you repeat the same assay a few years later in a different nested case control sub-cohort. By contrast, doing cohort-wide assays, they're very expensive in the short term because anything times 500,000 is a lot of money. But in the long term, they're very cost effective because you're only doing it once. You're minimizing sample depletion. You can massively improve your quality control of the assay because you're doing it at one time. It supports different comparisons of the biomarker across a whole variety of different diseases, and the data is widely shareable, and you can enable a really broad range of research. So I just wanted to show you an example of the use of the metabolomics data that we have on all 500,000 participants, and how that's being used to help stratify populations for precision medicine. So just like the polygenic risk score idea, researchers here from Nightingale Health have taken exactly the same approach to identify individuals in the highest decile of risk based on their metabolomic profile. So if you take all of the metabolites across an individual, those who are at highest risk of developing certain diseases. And you can see here that those in the top 10th of risk profile have a much higher risk of developing a given condition compared to the rest of the population. Now, if you can see here, if you combine the polygenic risk score with the metabolomic risk score, that may improve risk prediction of diseases over and above one another. So if you just take myocardial infarction as an example, you can see that the first dot in pink, you can see is the polygenic risk score. So there's about those in the highest polygenic decile, polygenic risk, have about a 2.5-fold risk of developing MI. If you look at the metabolic risk score, which is in purple, they have about a 3-fold increased risk. If you take the two together, you can see that that improves the risk prediction even further. So it's a really good demonstration of how taking the genetic data and the metabolomic data combined, you can help improve risk prediction for populations for a whole range of different diseases. So moving on to proteomics, this was done by a collaboration, again, of about 13 different pharmaceutical companies who got together to invest in UK Biobank to perform proteomics in about 57,000 individuals using the Olink platform. And we've just released data at the end of last year on those 3,000 circulating proteins. And that sample included randomly selected participants, plus some augmented with certain diseases. And various publications have been produced from this, notably this one in Nature at the end of last year, which looked at the genetic association of variants between these 3,000 proteins. And this data already is the largest collection of genetic associations with protein expressions, or what we call pQTLs, with 10,000 genetic associations being found, the vast majority of which were novel. So early analyses on the proteomics data has focused on, can we use protein levels as early disease detection biomarkers? And what's the association of protein expression with the risk of incident disease and mortality? So those are kind of the early analyses that are underway with the proteomics. We would love to be able to scale this up to the full cohort. It's just a question of finding out how we can fund this, because all of this work is hugely expensive. So moving on from kind of the biomarkers that we've really focused on over the last 5 years, we've also been focused on bringing participants back to an imaging study of up to 100,000 UK Biobank participants. So this is the largest ever performed study by some margin, actually, of imaging in a research study on a cohort-wide basis. So by the end of this year, we hope to have imaged 100,000 participants. We've already done 80,000 of them across 4 dedicated imaging centres across the UK, using highly standardised protocols, MRI of the brain, of the heart, of the body, and a carotid ultrasound and a full body DEXA scan, plus largely a repeat of what they did at recruitment. So we have repeated measures on individuals. And what's interesting is that once you have MRI scans at this scale, you very rapidly realize you need very quick automated processing pipelines to generate derived phenotypes from these scans. So for example, from the brain MRI scan, we now have over 3,000 metrics available, ranging from brain volume to volume of the hippocampus and so on and so forth, that researchers can use so you don't have to be experts in imaging to be able to use these data, because you can use these derived phenotypes based on the automated algorithms. So we've now just started a repeat of this imaging assessment, because there was a very strong case that actually having one image scan at one time was great, but what would be of real scientific added value was if you could look at change over time in internal pathophysiology. So obviously, you know, we're changing all the time, we're getting older, we're getting diseases, so we really need to be able to measure that. So we've just started a repeat imaging assessment of 60,000 individuals. which will go on for most of the rest of this decade. 7,000 have already come back for their second scan. This is being funded by Calico, MRC, which is basically from the UK government, and also CZI. And what this will enable researchers to do is to look at changes in imaging phenotypes over about a 2 to 7 year period during middle and older age. So you can look at, for example, what are the determinants of bone loss? What are the determinants of changes in the structure and the connections in the brain over time? And it will also enable researchers to identify early biomarkers based on the imaging scans that are then related to subsequent diseases. So they're really good intermediate phenotypes. So for example, can you identify changes in the structure or the function of the brain that precede cognitive decline or that precede a diagnosis of dementia? So this is the only study in the world where it enables this type of research to be performed. So in addition to repeating the imaging study, what we would love to be able to do over the next few years is to try to bring back as many of the surviving participants as we can to do a repeat of the baseline assessment that they did about 20 years ago at recruitment. And whilst we've got people at the assessment centres, it also provides us with a fantastic opportunity to include additional assessments related to aging and frailty. I mean, the age of our cohort now ranges from mid-50s to mid-80s. The average age is about 70. So now is a wonderful time to be able to measure strength, balance, movement. What are the early biomarkers of Parkinson's, dementia, other neuro diseases? Can we measure cognitive function and memory and vocabulary? Things we haven't done before, but are really relevant now to this aging population. So obviously having assessment of change from recruitment 20 years ago to now, change in lifestyle, change in physical measures, change in biomarkers would be hugely important, as change is much more strongly associated with disease than just having one measure at a time. And also enable you to look at changes in functional outcomes or intermediate phenotypes. So changes in cognition, changes in blood pressure, changes in cholesterol, as an example. So moving on to, the number of health outcomes that we've identified in the UK Biobank resource so far, we have about 15 years of follow-up through cohort-wide linkage to the National Health Service electronic care record. So unlike the US, we're quite lucky in this country in that we have this wonderful thing called the National Health Service, where some electronic health records are centralized. So, for example, we have a national death registry, a national cancer registry, and also our hospital admissions data are also centralized in one place. It's separate in England, Wales, and Scotland, but we don't have to go to individual states or individual hospitals to get those data. So it's relatively easy for us to be able to link all of our 500,000 participants to death certificates, cancer registrations, and inpatient hospital admissions, which gives us information on diagnoses in hospital, operations and procedures they may have that go back to the 1980s. So we have this information over a long period of time. We've also, since the pandemic, also got COVID testing data and so on. In terms of primary care data, so this is data when you go to your physician for something that's pretty mild. There's so many conditions that are only managed in primary care. So if you think of things like diabetes, depression, osteoarthritis, migraines, eczema, all of these minor conditions that cause a huge amount of days off sick and morbidity, but are not severe enough to make it into hospital, these are dealt in primary care in the UK. Now, that I could go on for a whole extra hour about the problems we've been having on linking to primary care data. We have it for just under half the cohort up until about 6 years ago. Since then, it stopped for various governance reasons, and we still, to this day, cannot link to primary care records, even though we have explicit consent from all of our participants to link to their medical records. So it's caused a huge frustration for us, but nonetheless, even without the primary care data, what you can see here from this table is that we have complete follow-up until the end of 2021. You can see there's already thousands of incident cases of the most common diseases in the resource. And over the next 5-10 years, that number is just going to accelerate for some diseases on an exponential scale, given the age range of the cohort. So already you've got huge statistical power to do really interesting granular research into some of these conditions. So one of the things, I think the most defining feature, if I'm honest about it, for UK Biobank, it's not all the fancy data we have. It's not the length of time that we've been able to follow up our participants. It's not even about the quality of the data. It's the fact that we've made these data really easy to use by researchers worldwide. And that is the reason UK Biobank has been it's been so successful and has generated so much extra funding since the project started. When the project started 20 years ago, it was a wild experiment. Nobody knew how it would develop because nobody had developed a resource that from the outset was aimed to be easily accessible by academic and commercial researchers worldwide on exactly the same basis. So nobody had done that before. So it was a punt in the dark. It really was. But what we found is that once you made the data accessible to researchers in a transparent, easy, cost-effective way, then researchers will come and use your data. And you can see from the graph on the left, we've got now over 40,000 researchers who are registered to use the resource across academia and commercial. The US actually, there's more US researchers who are using UK Biobank than any other country in the world, even in the UK. When we opened up to the research community in 2012, it was mainly UK academic researchers who were accessing the data because they helped us build the resource. Once we'd released the genotyping data in 2017, then overnight, it kind of the floodgates opened, and now it's the US are the top users of the resource, the UK followed by China. And it's being used all over the world, around 100 countries are accessing UK Biobank data now for a whole range of research. And there's now over, as you can see here on the graph on the right, exponential increase in number of publications and citations using the resource, over 9,000. I think the latest is actually 10,000 publications now. And on average, it's between 10 and 12 papers a day published using the resource. And to be honest with you, it's really hard to keep track of the output and the sheer breadth of research that's being done. But I can't emphasize enough just how it's this criterion about the resource that has really made UK Biobank so special. So in order to democratize access to the resource even more, we launched our secure online research analysis platform in 2021 because we have 24 petabytes of data with the whole genome sequencing data. You cannot shift that data around at all. So we launched this platform in 2021 as part of the whole genome sequencing project. So this is a platform where you can go, all you need is an internet connection. You go online, you can download your project into a workspace, into the cloud, do all of your analyses in situ using their tools available on the platform, or you can even ingest your own tools and other data sets into your workspace if you want to do pooled analyses and so on. And Amazon Web Services have also donated research credits. So if you're an early career researcher, or you're a researcher from a low middle income country, you can essentially access the data for free on the platform. So there's no cost to accessing the data, and there's no compute cost to analyze the data. And this has helped to democratize access to the resource. And we're finding that these bright young early career researchers who perhaps could not access UK Biobank before because it was too expensive, they didn't have the supercomputers in their universities or their institutions, can now do so because of the online analysis platform. And if any of you are using UK Biobank on the platform, I'd love to hear your feedback on the usability of it, because we constantly are trying to improve the usability for its users. So just to kind of finish off, I think UK Biobank, you can summarize it it's a combination of four dimensions. So the first is its large scale, 500,000 individuals across, age, different ethnicities, although individuals were mostly white, 5% ethnic minorities, different socioeconomic circumstances, different geographies, and so on, when they joined the study 15 years ago. High engagement of participants in the study, only about 1,000 have withdrawn from the study since recruitment. So we have a very engaged, altruistic cohort. The second unique characteristic, I think, is depth. We have really granular detail about exposure to lifestyle factors, environmental linkages, medical history data, combined with all of the range of biomedical assays I've spoken about, the genetics, as well now multimodal imaging data, also accelerometry data from Fitbit type devices that we've given out to 100,000 individuals. The third aspect is the duration of the study. So we have just over 15 years of complete follow-up now of our whole cohort. And as I've shown, there are already thousands of incident cases of the most common health outcomes to enable you to do really robust science into the determinants of these conditions and help find ways to prevent and treat them. And the fourth thing is, of course, accessibility. It's there's a rapidly growing number of researchers worldwide who are now using the resource for a whole range of different types of research and discovery science. So there are over 2,000 papers published last year alone. I mean, this is just a smattering of publications I found in the last couple of months that really kind of just showcase the breadth of research that's being done. So, ranging from association of diet and bowel cancer, what's the association between social isolation and mortality, looking at air pollution, how that combines with genetics in relation to lung cancer. And this article in the middle was a really interesting paper taking the physical activity monitor data. So this is data from a Fitbit type device that we gave to 100,000 participants about 10 years ago to wear for 7 days. And these researchers found that data from that device could be used to accurately predict Parkinson's disease 7 years before they were diagnosed. So really interesting use cases of the types of research that you can do using these data. So I just want to end by saying UK Biobank is absolutely not the panacea for every type of research question. So we can't answer everything, particularly if you're interested in looking at how associations differ between different ethnic groups. You really need large-scale population studies strategically located in other parts of the world that have, wide variability in terms of ethnic diversity, genetic diversity, social circumstances, exposures to lifestyle factors, and of course, environment. So what we need and what it does exist, but I think there needs to be more strategy around it, is a global ecosystem of large-scale, population-based cohort studies that are easily accessible to the global research community, so we can do pooled analyses to do this discovery science, replication science, to really improve health for all populations, not just the white population. And of course, this is part and parcel of what the International 100,000K Consortium, Cohort Consortium does. But I really do want to emphasize the point that, you know, there are still gaps in the world. You know, we don't have anything in India, not large scale. We don't have a large scale cohort in Russia. There's no large scale cohort in large parts of Africa. So, you know, I think if we can come together and really try to fund large-scale population-based cohorts in strategic parts of the world and make those data shared by the worldwide research community, then we're really onto a winner, I think, in terms of being able to improve public health. So I just want to end by acknowledging obviously all the staff at UK Biobank who work really hard to keep the wheels on the train, but actually, obviously our funders, but the most important people to thank, of course, are our 500,000 participants, without whom none of this would be possible, and they continue to be our strongest advocates and continue to give us all the information they can about themselves.